This video will instruct you how to dissect sheep brain in the laboratory. Sheep brains that we will use in lab are preserved in formaldehyde. For the brain dissection, you will use scissors, forceps, and a knife. When dissecting the brain, please preserve the dissected brain parts in a cyan plastic bag while dispose the membranes in the waste plastic bag. The first thing you will observe when you're looking at the brain is a thick membrane that looks like a swimming cap. That membrane, called dura mater, gives protection to the brain. Below dura are arachnoidea, net-like structure, and a thin membrane called pia that closely attaches to the brain. We will first cut dura with the scissors in the middle of the brain and remove from cerebral hemispheres in cerebellum. We have to be very careful when removing dura from the diencephalon and brainstem, since we want to preserve as much as we can cranial nerves and pituitary gland that are on the basal aspect of the brain. In sheep brain dissection lab 1, we will observe sheep brain gross morphology on the superior view, inferior view, and mid-sagittal view. During lab, you will identify adroit structures visible at each view. On the superior view, we will identify frontal lobe, parietal lobe, temporal lobe, and occipital lobe. We will also identify sulci and gyri. On the inferior brain, we will identify cerebral hemispheres, diencephalon, optic nerve, optic chiasm, optic tract. Here also you can see this hole here, it's called infundibulum. So pituitary gland, which is attached to the diencephalon, is attaching through infundibulum to the rest of the brain. So below diencephalon is midbrain. Then here we can see pons, medulla oblongata, spinal cord, cerebellum. We will now cut brain into two equal halves, left and right. That is called sagittal section of the brain. Two halves of the brain are connected through the structure called corpus callosum, which is white matter of the brain. And when we cut through the brain in this way, we will cut through corpus callosum. If we cut into two equal halves, left and right, that is called mid-sagittal sectioning. On the mid-sagittal section, we will identify cerebral hemispheres, corpus callosum, lateral ventricle, which is underneath. Then we have thalamus here, which is part of the diencephalon. So this is hypothalamus. This is epithalamus or pineal gland. Superior colliculi and inferior colliculi, which are the part of the midbrain. This part here is pons, medulla oblongata, and this is spinal cord. So here we can see cerebellum with gray matter and white matter. This concludes sheep brain dissection.